We've talked a little bit about the overall function of the renal system and how urine is made. We're now going to go into some of the anatomy and physiology of the kidneys and really go into what the function of the kidneys is specifically in making urine. So first of all, the location of the kidneys, they are retroperitoneal, meaning that they are behind the peritoneum. You'll find that the right kidney is slightly lower than the left. This is because on your right side you have your liver that's taking up a lot of space and displacing the kidney. The external anatomy of the kidney, you'll notice that it has got three distinct locations outside of it. So you have the renal fascia. This is the outer layer of dense fibrous connective tissue that anchors the kidney to the adrenal gland and or anchors the kidney and the adrenal gland to the surrounding structures. You have the pararenal fat capsule. So this is the fatty mass that surrounds the kidney, which will help cushion it against blows. And you have the fibrous capsule. This is going to be the innermost region that is preventing infections in the surrounding area. So if we look at the kidney itself, you'll notice that at the central concave region of the kidney, you have a lot of different structures that are entering into the kidney. This is what is known as the renal ileum. On the outer surface or outer portion of the kidney, you have what is known as the renal cortex. Deep to that, you have the renal medulla. Within the renal medulla, you find renal pyramids. So these are cone-shaped masses. They consist of parallel nephron loops and the collecting ducts. The apex of the renal pyramids is what we call these things the renal papillae. And then the renal columns, these are extensions of the cortical tissue that is really going in between the renal pyramids within the medulla. All right, if we look at the structures that are going out of the kidneys, you'll notice that we have several minor calyces. So those are those small funnel-shaped things that are coming from the renal papillae. This is really going to be where you are collecting a lot of the urine initially. These are going to merge into what is known as the major calyces. So these, there are two of the three major calyces that come together, ultimately forming the renal pelvis, which is going to be continuous with the ureter that leaves the ileum. All right, if we look into the flow of blood as it goes through the kidneys, you'll notice that the aorta is going to flow down into the renal artery, which is going to divide into segmental arteries. The segmental arteries are going to divide then into these interlobal arteries that go between the lobes. Then you have the arcuate arteries that are going to extend from the interlobal arteries. Then finally, coming from those, you have the cortical radiate arteries. These then lead to the afferent arterioles. So if we zoom in on these, you'll notice the afferent arteriole is going to lead into the glomerulus. The glomerulus is going to then have some of the arteries exit as the efferent arterioles that then go into the peritubular capillaries. So this is going to be where you initially filter the blood. So the filtration is going to happen over here with the glomerulus, but then it divides and goes around these tubules there, the secretion and reabsorption is going on before finally going back to the cortical radiate vein. So let's go back here. So you have the afferent arteriole going to the glomerulus and the capillaries associated with that going to the afferent arteriole. Then that leads to the peritubular capillaries, or the vasa recta. So the vasa recta is a specialized hairpin-shaped blood vessels in the kidney that run parallel to the loops of Henle. The cortical radiate vein is then going to collect the stuff from the peritubular capillaries, taking it to the arcuate vein. You'll notice that some of the peritubular capillaries are going to go directly to the arcuate vein as well. These then will join at the interlobal vein before going to the renal vein that drains into the inferior vena cava. Notice that we talked about a system here where you have an afferent arteriole leading to an efferent arteriole. So and in between there you have a capillary bed. So this is the definition of a portal system. However, this is unique in that it is the arterioles that are found between the two capillary beds. Other portal systems, like you find in the hypothalamus going to the anterior pituitary, or the blood vessels of the digestive system going to the viscera of the liver, these are going to be venous in nature. So it's going to be veins that are connecting the two capillaries in those instances. All right, so let's talk a little bit about the nephrons. So the nephrons are the functional units of the kidneys. They're going to take a filtrate of the blood and modify it into urine. So we have the renal corpuscle. That is going to be made up of the glomerular capsule. The glomerular capsule is where the filtrate is initially being made. It's going to lead into the proximal convoluted tubule. So that is everything that's coming over here. It's convoluted because it's winding. So here we're going to talk about forming urine. That's everything that's happening after the filtrate has happened until it is modified into what is true urine. From the proximal convoluted tubule, it'll go down the descending loop of Henle and go back up the ascending loop of Henle before it goes to the distal convoluted tubule, and then finally ends in the collecting duct. So let's take a look at the renal corpuscle. So you have a capillary here that is surrounded by these podocyte cell bodies. The podocyte cell bodies are uniquely shaped that cover the glomerular capillaries. These form filtration slits, which are going to ultimately leave small gaps that form a sieve. So these pedicels are the links that are going between, so you have the podocyte bodies, the pedicels are the extensions coming from them, and then you can see the filtration slits being where the filtration is happening. So blood passes through here, and 10 to 20% of the plasma filters between these filters where it is funneled to the PCT, the proximal convoluted tubule. If you look at the capillaries, they have the basement membrane, the endothelium, and tight junctions with fenestrations that are serving as windows for things to happen, to go through. So the fenestrations prevent filtration of blood cells or large proteins, but do allow most other constituents through. These fenestrations let things cross if they're less than 4 nanometers in size, and if it's even smaller than that, less than 8, and if it's a little bit larger, 8 nanometers, it can still pass. Electric charge can also affect the ability of things to cross the membrane, since things that are charged will tend to be repelled by the membranes that are here, making it harder for things to get through. All right, so lying just outside of Bowman's capsule and the glomerulus is what is known as the justoglomerular apparatus, or the JGA. Here you can see the podocytes that are surrounding the capillaries. Between the entrant, the efferent, and the afferent tubules, you have the justiglomerular cells of the justiglomerular capsule. And then at this initial part, the DCT is going to come in contact with the macula densa. So the distal convoluted tubule has these cells that surround the macula densa that monitor fluid composition of what's going through the DCT. Thus, in response to the concentration of things, especially like sodium flowing past them, these cells can release paracrine signals, which will influence how much is being filtered present there. So the macula densa sensing sodium and acting as a paracrine signal to the cells surrounding it. The justiglomerular cell is a modified smooth muscle cell that is going to respond to signals from here and contract or relax depending on it, so that you have more or less filtration occurring accordingly. All right, so let's talk about the second function of the macula densa, which is to regulate renin release from the justiglomerular cells of the afferent arteriole. So active renin is a protein that is comprised of about 304 amino acids. It can cleave into several peptides or amino acids the protein cleaves several amino acids from angiotensin agin to form angiotensin 1. So you have the liver cell releasing the angiotensinogen. You have the renin that's causing the enzyme reaction to form angiotensin 1. Then in the lungs, Then in the lungs, you have angiotensin converting enzyme that's going to convert angiotensin 1 to angiotensin 2. This is going to then go throughout the body and stimulate widespread vasoconstriction, as well going to the adrenal cortex that causes aldosterone release. Aldosterone stimulates sodium uptake in the apical cell membrane in the DCT and the collecting ducts. And then ADH causes aquaporins to move to the collecting duct plasma membrane, which is going to increase water reabsorption so that you get more water being reabsorbed back into the body, and ultimately increases blood pressure as a consequence of that. You may have heard the term diuretic. So this is a compound that is going to increase urine volume. Caffeine and coffee and tea work by promoting vasodilation in the nephron, increasing this overall glomerular filtration rate. Alcohol can also increase the glomerular filtration rate, but it does this by inhibiting ADH release and not by promoting vasodilation in the nephron directly. All right, the proximal convoluted tubule is going to be what collects fluid from Bowman's capsule. The cells here are made up of simple cuboidal cells leading down to the nephron loop into the distant collecting tubule. So we're going to talk now about what is going on at each of these. The proximal convoluted tubule, here you're going to be having things such as glucose, amino acids, proteins, vitamins, lactate, urea, and uric acid being reabsorbed back into the blood. And then ions like sodium, potassium, calcium, magnesium, bicarbonate, chloride, and water are also going to be reabsorbed. The urea, uric acid, creatinine, and certain drugs do get secreted into the PCT here as well. and you do have H plus and NH4 plus ions being pushed in there. So the glucose amino acids and proteins, they are going to be reabsorbed using Symport with sodium. So as sodium gets reabsorbed, so are these other contents. In the loop of Henle, you have a thick portion and a thin portion. So the thin portion is going to consist of simple squamous epithelium, while the thick portion is going to consist of simple cuboidal cells. In the thin portion, you're going to have water being reabsorbed, while urea is going to be secreted into it. And then in the thick portion, you're going to have ions like sodium, potassium, and chloride being reabsorbed. Finally, in the distal convoluted tubule, made up of more simple cuboidal epithelium, you're going to have the active secretion of sodium, chloride, bicarbonate ion, and water. So these are going to be pumped against their concentration gradient, meaning that you need to have active transport and thus need ATP and the mitochondria. You have active secretion of potassium ions, ammonium ions, and H+. Ultimately, by the time you go into the DCT, you've had about 80% of the water recovered, with the remaining being collected by the collecting ducts, which will collect about 10 to 15, and so you only have about 5%. This can vary depending on a number of different things. So in the collecting ducts, you are going to have water and urea. Now the amount of aquaporins in the collecting ducts will determine how much water gets reabsorbed. This is largely going to be a product of how much signaling you have from the aldosterone that's signaling for the return of aquaporins or the removal of aquaporins in the collecting duct. All right, so we look at the rate of glomerular filtration as a way of monitoring health. So renal blood flow, you are going to have about 1050 milliliters per minute going through here. The renal plasma flow is going to be about 578, and then your normal glomerular filtration rate is about 110 milliliters per minute. Urine, you form about 1 liter to 2 liters per day if everything is normal and you're staying hydrated in the same way. The reason that this has all happened have to do with the colloid and hydrostatic pressure that the blood has. So the colloid osmotic pressure is drawing water back into the blood, while the hydrostatic pressure pushes it into the capsule. And so the difference, or the capsular hydrostatic pressure is about 15 millimeters. So you have a net outward pressure of about 10. So you notice the 30 plus 15 is 45. It's about 10 less than the blood hydrostatic pressure, which leads to about some net filtration occurring here. Most substances move across the PCT more substances move across the PCT than any other part of the nephron. Symport, I mentioned earlier, this is when you have one ion moving one way and the other ion also moving in that same direction or in the opposite direction to move them. So antiport is going to be when it moves in the opposite direction. Symport is going to be when it moves in the same direction. One thing that's very important is the counter current multiplier system. So the descending and ascending loops are next to each other and their fluid flows in opposite directions. So this leads to a action of actively pulling the concentrations of urea and sodium deep in the medulla. thus acting as a countercurrent exchanger. So notice that the interstitial osmolality is increasing as you go down here. Water is going out and urea is going in. So here you're having the osmolarity increase, but here you're having sodium and chloride going out and water staying in. So water stays in here, but it's drawn out here, thus affecting the overall pull of sodium, since sodium will move out here as it is pulled across. All right, so we're actually going to stop there. That basically goes over the kidney and its function. Next video, we'll talk about the rest of the path that the urine takes as it exits the body.